everyone, my name is Sonali. Welcome to the channel Success EduTech. Today we are going to learn about plant growth regulators or plant growth hormones. Please watch the complete video and please like, share, subscribe to our channel. Please watch previous videos also. Link is given in the description. We will see what is meant by plant growth hormones. Plant growth hormones was coined first by Starling in 1906 in animal physiology. The internal factors that influence the growth are known as growth hormones or growth regulators. Just like this, from seedling and then crop is growing due to the growth hormone. Now, plant growth regulators can be broadly divided into two groups based on their functions in a living plant body. So plant growth regulators are nothing but growth promoters which can help for the growth in plants, and growth inhibitors which can inhibit the growth. So growth promoters are cell division, cell enlargement. These are all the process including this growth promoter. Pattern formation, tropic growth, flowering, fruiting, seed formation. So these plant growth regulators are known as growth. Growth inhibitors is the dormancy, means the inactivity of the parts of plants, and abscission means detachment of the parts of plants, that is dead leaves or ripened fruit, etc. The plant growth regulators of the other group play an important role. Another function of this PGR is response to the wound and traces of biotic and abiotic origin. Means these PGR, plant growth regulators, can save the plants from the traces of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. So PGR, plant growth regulator, is abscisic acid, helps in the in inhibiting activities, and ethylene is the gases which also help in these growth activities, which also helps for the inhibitor of growth activities, that it can ripen the fruits. Types of plant growth regulators are auxin, then gibberellins, then cytokinins, then ethylene, and last one is abscisic acid. So these are the 5 natural growth regulators. Now, growth regulators and their chemical composition. So which are the growth regulators? That is auxin, help in growth promoters, that is indole compounds, and that is indole 3-acetic acid IAA or indole butyric acid IBA. Cytokinin, that is kinetin, these are the names for fural amino purine. Abscisic acid, that is derivatives of carotenoids. Gibberellic acid, that is diterpenes, and last one ethylene, that is the gaseous. The observation of Charles Darwin and his son Francis Darwin reported that the calyptiles of canary grass, means the sheath of protecting the young shoot tip of canary grass, responded to unilateral illumination by growing towards the light source, means the growth of plant towards the direction of the light. For example, see this in picture, and this is also the direction of growth of plant towards the light. After a series of experiments, it was concluded that the tip of calyptile was a site of transmittable influence that caused the bending of entire calyptile of oat seedling, like this. Auxin can promote this growth. It is a bending part. Auxin was isolated by, now this auxin hormone was isolated by F.W. Venn from tips of calyptile of oat seedling. Okay, means from the tips of shoot, young shoot tip. It was first isolated from human urine also. See, this is the calyptile and seminal root, and this is the radicle. So in plant from this, and in animal physiology, this is a urine, human urine. The term auxin is applied to the IAA, indole acetic acid. It is the example. and another natural and synthetic compound having certain growth regulating properties. It consists the growth regulating properties. They are generally produced by growing apices of the stem, apices of stem and roots from where they migrate to the region of their action. So this is the apices of the stem and this is the apices of the roots. So the growth can happen from this towards this. Auxins like IAA and indole butyric acid IBA have been isolated from plants, that is natural auxin. So this is auxin isolated from the plant. This is the ring structure indole 3 acetic acid formula, and this is the total structure of IBA. This is butyric acid. Natural auxin, both are the natural auxin. Naphthalene acetic acid, this is structure of naphthalene, double ring structure, and D4D dichlorophenoxyacetic acid. And these both are synthetic auxin. Now, all these auxin have been used extensively in agricultural and horticultural practices. This auxin hormone is useful in agricultural and horticulture practices. They help to initiate rooting in stem cutting and application widely used for plant propagation. In rooting also and in plant propagation also. See in this picture, it is the cutting portion, then it is just like a dip in water or tissue culture, and this is the technique of the formation of the seedling. Auxin promote flowering. Due to the help of auxin, flower can form. So it promote the flowering, for example, pineapple. Now, they help to prevent fruits and leaf drop, detachment of the leaf and fruit. They help to prevent the detachment of help and fruit from the plant at early stages, but promote the abscission of older mature leaves and fruit. When the leaves and fruit get mature and it will be old, that time abscission process is necessary, which happen due to the abscisic acid. See abscisic zone and leaf scar. In most higher plants, the growing apical bud inhibit the growth of lateral axillary buds, a phenomenon called apical dominance. This is apical bud and this is lateral bud. This is apical bud. So in most higher plant, the growing apical bud inhibits the growth of lateral bud. It inhibits it. The growth happen here and it inhibits. So this phenomenon is known as apical dominance. Removal of shoot tips, that is decapitation, usually result in growth of lateral buds. So removal of shoot tip, these usually result in growth of lateral buds. So it can, if we remove this, it can help in growth of lateral buds. So this is the lateral shoots which can grow from lateral buds. It is widely applied in tea plantation and hedge making like this. And is tea plantation. Auxin also induce parthenocarpy. Example, tomatoes. Means it can form a seedless fruit like tomatoes, banana. Auxin also controls xylem differentiation and helps in cell division. This is xylem differentiation and helps in cell division also. They are widely used as herbicides. Herbicides means it can spray on the herbs. 2,4-D, widely used to kill dicotyledonous weeds, does not affect mature monocotyledonous plants. 2,4-D is used for dicotyledonous weeds, not for monocotyledonous. And it is used to prepare weed-free lawns by gardener also. So this is the 2,4-D, which is amine weed kill. Now the MCQs related to this topic. Phytohormones are means the plant hormones are compound that control various physiological functions in plant. Plant growth regulators, special enzyme produced by plants, and both 1 and 2. So the answer is both 1 and 2. Now, a vena curvature test was performed by discovery of gibberellins, auxins, cytokinins, and ethylene. And the answer is auxins. The naturally occurring auxins are NAA, 2,4-D, IAA, and both 1 and 2. Answer is IA. Thank you very much, students. In the next video, we will see about plant growth regulators, that is gibberellins, cytokinins, ethylene, and abscisic acid. Thanks for watching.